So let's look at electrical activity in terms of automaticity. And so this is where the heart contracts rhythmically as a result of action potentials that it generates by itself. Okay, and so this has to do with that autonomic function. Okay, so contractile cells and autorhythmic cells are going to be the two types of cells that we find within the heart. So 99% of the cardiac muscle cells do the mechanical work of pumping, and then less than 1% are going to be these autorhythmic cells that are going to initiate and conduct action potentials that are responsible for contraction of working cells. And so this is where we have the different pacemaker potentials in autorhythmic cells that are complex interactions of several different ionic mechanisms with calcium, potassium, and sodium. All right, so this allows for excitable cells, just like in muscle, skeletal muscle, and neurons, and then so cardiomyocytes, okay? So the big thing for the autorhythmic cells is going to be the concentration of ions inside and outside of the cell, so very similar to what we covered with the neurons. And then also permeability of the cell membrane to these ions due to different channels, and then also differences of electrogenic pumps. So sodium, potassium, ATPase pump is going to be one that we'll see again. And also calcium transport pumps that maintain ion concentrations. Because again, as I said in earlier, calcium is going to be a big factor for this. The action potentials of autorhythmic cells now will be a little bit different than what we've seen with the action potentials in neurons. Okay, so first thing is a big difference that we find is that the threshold potential, instead of being at minus 55, we're now at minus 40. Okay, and also there's no true resting membrane potential because action potentials are self-induced and they're continuous. Okay, so the first half of the pacemaker potential is due to closure of potassium channels, and then whereas the second half is going to be due to opening of these transient type calcium channels. So we have two things going on. So we have closing of potassium and then also an opening of the short-lasting transient potassium or calcium channels. All right, then the second phase of depolarization that we have now are the opening of voltage-gated, long-lasting calcium channels. And so this allows for an increased amount of calcium to come in, okay? So then once we have the channels closing at about 0 to 10 millivolts, then we have an opening of the potassium channels, and so now potassium is coming out of the cell, okay? And so here what we find is that sodium actually does not play a major role. So the big change that we have are calcium coming in and potassium coming out. And so one of the mechanisms that we have though is a slow depolarization or pacemaker potential. And so this allows for our cardiac cells to not undergo summation or tetanus like it does with our skeletal muscle. So what we have too is the bottoming out is about negative 60 and then driftward up to negative 40. So this is about the threshold potential. So these are termed to be myogenic because these can reach threshold by itself. We don't necessarily need to have a neuron firing for it to induce an action potential. Now, these reaching up to threshold potential is due to the slow leakage of potassium coming out, but then there's also constant leakage of sodium coming in. And so this causes a slow depolarization, okay? So calcium channels are going to open, and then this is going to cause a rapid type of calcium to come in due to those L-type channels opening, okay? And then once we have hit our peak, then we have slow potassium channels that are going to open, and then this is going to have potassium coming out of the cell and then repolarizing it back to negative 60 degrees, or negative 60 millivolts, sorry. the location of the autorhythmic cells. So these are going to help control our heartbeat and are due to cardiac electrical signals. And so this is going to be the electrical conduction pathway, and so this is going to be initiated at the SA node or sinoatrial node. And this is myogenic, so again, self-inducing, where it has about 70 to 80 action potentials per minute. So the depolarization, and this is considered to be our pacemaker cells. And so the depolarization then is going to spread through the atria via gap junctions, and then also we find this within the right atria, and so this is going to spread into the left atria by internodal pathways. Okay, and then that will go to then the next cluster, which is the AV node or atrioventricular node. Okay, and so the AV node, there is going to be a slight delay for that signal from the SA node to the AV node to occur. And this is due to the slower formation of action potentials, and then it also, though, helps to allow for emptying of that atria to occur. Action potentials are going to travel then down from the AV node down the interventricular septum, and this is termed into the atrioventricular bundle or the bundle of His, which splits into a left and right atrioventricular bundle. Okay. And so from there, what we have are the Purkinje fibers that are going to wrap around the walls of both ventricles so that they allow for those cells to contract all the way around the ventricle. The Purkinje fibers or Purkinje cells are also going to conduct pulses very rapidly, and so this causes the cells of the apex, so down here, to contract simultaneously as they are towards the base as well. And so this allows for ventricular injection of blood that will go on into their corresponding vessels. So sympathetic activity of the autonomic nervous system can influence our pacemaker cells, and so we've seen this before where we talked about how norepinephrine and epinephrine can increase the amount or the rate of the heart and also the contraction of the heart too. And so these are going to bind to the beta receptors, so beta 1 have adrenergic receptors on the SA node, and then there's also beta 2 that are going to be on the AV node. Okay, and so they're bind to both the atria and the ventricles as well, so it's not going to be just on one or the other, which is different with the parasympathetic division. Okay, and so what this does then is norepinephrine and epinephrine causes a rapid pacemaker potential, faster action potentials. So what all in all that does is it increases heart rate, increases contractility, and conduction of these action potentials. So here in this graph, what we have are in the gray, these are normal, slow, if we're not induced with the sympathetic nervous system. And then here is going to be a lot faster. And so here we have an increase in heart rate. But note too, there's no increase in the action potential in terms of voltage themselves, right? It's just the rate or the frequency at which we're seeing it. Parasympathetic division, however, is going to have opposite effects. So here we're going to have acetylcholine, and it's going to bind to muscarinic acetylcholine receptors. So what this does is it increases potassium permeability and then decreases calcium permeability. And so this leads to a larger hyperpolarization of the membrane, and so it takes a longer time to reach threshold. Okay, and so that's going to slow the rate of the action potentials. Now, a big difference here is that it is going to terminate only on the atria, okay? So whereas the sympathetic can affect both the ventricle and the atria, here with the parasympathetic, it affects only the atria. So it's only affecting then the SA node, and then so the SA node is slowing down. And so the SA node is going to decrease the heart rate ultimately that's going to decrease that contractility because of that myogenic response, and then also we'll have a decrease in conduction of action potentials. So here in the blue, what we have is an example of the hyperpolarized state, and so becomes more negative, and then it's taking a longer time now to reach threshold. Again, though, note here, though, there's no difference in terms of the voltage change of the action potential, okay? All right, so then looking into how do we measure electrical activity of the heart? Well, this is done through an EKG or ECG, and you guys have done this in lab already. And so the electrical activity that's present in body fluids from the cardiac impulses that reaches the body's surface. And so we have these different leads that we can record the electrical activity through the heart. And so we have positive, negative, and grounds that we can incorporate. And so you only need to have 3 leads on the body in order to measure the electrical activity of the heart. And so lead 1 will be from the left to the right arm, and then lead 2 we have from the right arm to left leg, and then from left to left. And so what that allows for is we can look at depolarization that's occurring within the right and left atria, depolarization that's occurring within the left and right ventricle, or left atria and ventricle, and then lastly from the left and right, or right and right atria and ventricle. All right, so then what we have here is what an EKG looks like, okay? So here we have the SA node fires, and again, SA node is going to be our pacemaker potential. And so what that generates is going to be this P wave right here. And so the P wave is going to denote then depolarization of the atria. Then we have this segment called the PR segment, and so here in an EKG, the PR segment denotes the delay from the SA node to the AV node, and so that AV nodal delay. Our QRS segment is going to denote the ventricle depolarization. Okay, so here it's showing where it's going down the atrioventricular bundle and it's going to wrap around ththose Purkinje fibers, so around that ventricle. And so the atria, though, at the same time is going to be repolarizing because it's depolarized and now it wants to repolarize. So then we have the ST segment. And so this is the time in which the ventricles are contracting and are emptying their blood now. Then this T wave right here that we find is going to be where the ventricles now are repolarizing. And so again, they've depolarized, so now they want to repolarize. And again, has time in between. And so why we see within the EKG and within electrical activity is that slow reaching of threshold. And then lastly, we have this TP segment, and this is when the ventricles are relaxing and they're filling with blood again. And then so this will begin the process all over, and we have then P wave with atrial depolarization. And so here's another visual for looking at it. So here we have our T wave or P wave. So here we have P, and then we have our Q, then we have our R, S, and then here's our T wave here when we look at an EKG, and then here's the delay, and then the P wave, okay? So again, with the P wave, here we have the atria going from the SA node is being depolarized and spreading through both atria, okay? That's why we see a little bit of a delay because it has to go through both the left and right atria. And then we have the atrial depolarization is complete, and then so from the SA to the AV node, that AV nodal delay, we have then the depolarization occurring within the ventricles through that bundle of His and those Purkinje fibers that are going to wrap around the ventricles. And so then when that depolarization is completed, then we have contraction of those ventricles to allow for emptying, and then the ventricles again repolarize, and so that with repolarization what we have is the relaxing and filling again with blood, and so this will continue until it's completely filled within the ventricles.